Okay, now we've scared all those people away. Did want to talk about a couple of things from the exam. One of the things that I do is I'll go through and look at all the questions that the most people had trouble answering. And so the next 4 questions are the ones that at least 75% of the people missed. And I just wanted to go through those really briefly. Most of these, some people would say, are stamp collecting, the little minuscule details, but there are a couple of important ones that I did want to cover here. So the genes that seem to be the most necessary to maintain, since they're present in the smallest cellular genomes, are the translation protein genes, because translation seems to be the most fundamental aspect of biology as far as we know it that says your cells. And we'll get, next term I'll talk about this in terms of virology, because clearly viruses are the coolest things ever, and they've managed to not have to deal with these. But all viruses have to use cellular translation, and that's maybe one of the reasons that viruses are dependent on cells, is because all cells have translation. And when we talk a little bit about RNA and the RNA world and evolution at the very earliest times, translation was probably one of the very earliest things to happen. And so that's another reason why these translation proteins are incredibly well conserved in these genomes. Next one had to do with Chargaff versus Levine and Watson and Crick's models, sort of a combination of a whole bunch of different things in that one lecture. So the critical difference between Chargaff's experiments and those of Levine that allowed the development of Watson and Crick's model for the structure of DNA was that Chargaff used bacteria and Levine did not. And that's because all of the organisms that Levine looked at for DNA had very similar concentrations of C's, A's, T's, and G's, whereas some of the bacteria that they used, and in fact, Mycobacterium tuberculosis, that was one of the other questions I wasn't going to talk about today, but I talked about on D2L. By the way, there are lots of discussion of questions on D2L too. You have questions, take a look at those as well. The bacteria had a very high GC concentration, and so it's then very different from everything else that Levine had looked at. And so that's where Chargaff's rule came from, even though he never called it a rule. And Watson and Crick basically said, oh, it looks really close to 1. Chargaff said it looked close to 1 and wasn't sure what it meant. And Watson and Crick said, hey, it is 1. It's good enough, and we'll just use that. The next question actually had to do with the paper that I asked all of you to read. Which of the following were differences between Watson and Crick's model and that of Linus Pauling? Pauling's structure had 3 helices and the bases on the outside. But that just came from that paper. And evidently, either people didn't read it or they didn't read it in that detail to be able to remember it for the exam. The other question, and someone asked me this, in fact, maybe 1 or 2 people during the exam, what does PhD stand for, other than piled higher and deeper and Doctor of Philosophy? The answer is, I don't know what it stands for, and I was going to look it up before class, but I didn't have a chance to. I will by Monday. I will let you know. I think it stands for proline, histidine, and aspartic acid, but that's a guess. But what they do is they bind to methylated amino acid side chains. And I can claim Dr. Bartlett didn't do a good enough job of explaining this or went over it really, really fast. But the idea here is that it's these domains that bind to the different modifications. So even if you had no clue what the PhD domain stood for, we barely talked about ubiquitinated and phosphorylated histones. So it probably would have been acetylated or methylated. So 50% chance. But I guess, again, given the fact that more than 75% of the people missed it, in fact, in this case, it was more like 90% of people missed it, I guess that wasn't the way that people were thinking about it. And this is the slide, actually, that comes from. The PhD domain is this big gray thing right here. And there's your trimethylated lysine residue on the otherwise unmodified N-terminal tail of the histone. So, any more questions about that before we sort of move on? What everyone's been asking me about is this. Yeah, go ahead.
With the very first one, why is translation more
So the question is, why is translation more conserved than DNA replication? But replication in general. So I often say don't ask why questions in biology, but it's a great question, and I always have them on my exams. So I'm obfuscating here. What people think, and again, this is theory. We know that the translation proteins are conserved. Those are data. Those are nice, solid data. And so probably what happened, and again, I'm moving ahead a little bit into virology here, is that there probably was an RNA world where it was an RNA replicase. So RNA had both genetic activity but also the enzymatic activity. And then proteins arose, and then after that, much later, you got DNA. And so those DNA replication proteins, turns out that there's more diversity in DNA replication proteins in viruses than there are in cells. So a number of people, including me, think that viruses invented DNA. And so it actually came considerably later than translation, which was probably just RNA and then the, RNA going to protein. Then, okay, the next question is, why not transcription proteins? And we'll talk much more about the transcription proteins later, because you still have to go from RNA to protein. But that's DNA to RNA, so that you'd have to have the DNA first. So that's the hand-waving explanation for why that is. Again, until we have a time machine, we're not going to know, which may be a little while. Yeah, probably not, no. Okay, what people really care about, I guess, because that's what everyone's asking me about, approximate grades for the midterm. How do I do my grading? This is basically an excerpt, more or less directly out of my lecture 1 in the syllabus. I take the highest score on the exam. In this case, it was 44, after someone pointed out, thank you, that I screwed up on the keys. So 90% of that would be a score, not a percentage, but a score of 39. I'm sorry about D2L. D2L gives this percentage thing. I hate the percentage thing that it gives you in D2L. I don't think it's useful at all. So 39 and above, consider yourself having an A, 37 and above, A minus, et cetera, et cetera, et cetera, et cetera, et cetera. I don't give letter grades until the very, very end of the term. I add up all of your exam scores, don't drop any of them, and then normalize again to the high score. If that high score is not some ridiculous outlier by some anesthesiologist who happens to be sitting in this class for fun and messing up the curve. I had one of those in virology, by the way. Ended up being my PhD student. But I will look at that and go, there's clearly an outlier. And that actually is, to some extent, the case for this exam as well. There's one 44. I think there's 1 or 2 43s and a whole bunch of 41s. So this is, I would say, sort of the minimum that I would expect. It probably is going to end up being a little bit lower than that. But it's going to depend on how these people do on the other exams, et cetera. Or whether the anesthesiologist drops the class. Case may be. Okay, if you do have a score that is below 20, we should be talking. Yeah?
So my question is, so you'll normalize and then normalize again, so it won't normalize twice then for each exam?
No, so I only normalize at the end. I'm only going to normalize at the end of the term. So this is just so you have an idea where you stand. Because no one wants to see a number and go, oh, help, Dr. Stibman, where am I at? So.
It won't go down, it'll just go up.
Yeah, if anything, it would go up. And usually the scores on this first exam are the best scores for the midterms in terms of the high score. Okay, questions, comments, worries, freakouts, etc. Good. Okay, so let's move on. This stuff is now going to be on the next midterm. So we talked a bit about the actual mechanism of DNA replication before, but of course to get DNA replication to happen, you've got to start it somehow. And that's all about initiation. And this is a very general case of initiation here on the, I guess, right-hand side of the slide. You've got double-stranded DNA, and somehow the cell has to figure out where it's going to start. And so these are all what are called the origins of replication, just where things start. You have to get the 2 strands apart because the only way to get at the information is to pull those 2 strands apart. What does that? Helicases. And so getting the helicase on the DNA is the critical step in terms of getting an initiation of replication. So putting the helicase on. Getting those two strands apart, putting the helicase on. How does that happen? It's through the presence of these so-called initiator proteins, which are going to bind to the origin or nearby and get the helicase onto the DNA so that the DNA comes apart. As soon as it comes apart, you've got primases around that can make primers. You've then got polymerases, which can extend those strands. And as soon as you get far enough along in one of these replication forks, you can start making lagging strands as well as leading strands. A couple things to point out here. Since the DNA is double-stranded and symmetrical, from any origin, you're going to have two replication forks. So there's a replication fork on this end and a replication fork on this end. Sorry if it's a little hard to see from over here. And each of those will have a leading strand and a lagging strand. So a leading strand and a lagging strand. You're always going to have these two replication forks that what people also will say fire from an origin of replication. So yeah, then the main thing here is once you've got the two strands apart, then the rest happens more or less automatically. So how do you get this to happen? It's an initiator protein that binds near where the actual origin of replication is. And so when I say what is the exact origin of replication, that's going to be where that primer starts. And the primer is going to start on the leading strand. That's going to be exactly where your initiation is. Sorry, I may have missed your question.
If you go back to the last one, with the strands, the lagging strand of fork 2 catches up with the leading strand of fork 1, what would you call that?
So when the strands bang into each other,
And replaces
right.
the primer.
So it's going to replace the primer. At that point, you could even sort of thinking of this as being just an extension now of the leading strand for fork 1, because it will have cleaved up the primer and then attached to it. Now, I think the extension to your question, which we'll get to in just a second, is what happens when forks run into each other. And that's what we call termination. So that's, but basically here, once these guys then, and you're exactly right, it's the polymerase which will extend here, it'll hit the primer will get degraded just like it happens on the lagging strand. And then you'll have one, big long piece.
So you would, if you asked us, is this a leading strand or a lagging strand, you would say leading.
Yeah, well, so I'll ask about forks. At a fork, which would be here, then leading and lagging strands make sense. Somewhere in the middle, like this, where the lagging strand is hooked up with the leading strand, it's going to be really hard to say one versus another. Okay, other questions on that? Sorry. So again, these initiator proteins bind to DNA. The initiator proteins bind to specific DNA sequences. And so wherever you're going to get an origin of replication, next to it is going to be an initiator binding site. Then, right next to it is DNA, which is going to get pulled apart. Not terribly surprisingly, this is almost always DNA, which has a high percentage of AT base pairs. Why? Because AT base pairs are easy to pull apart. And then, once you've pulled them apart, then you put on the helicase, which is what this little orange loop here is supposed to be. And then once you got the helicase, then that's going to go and pull apart the two strands. Once the two strands are pulled apart, you have your primase, et cetera. Everything happens from there. So this is getting back a little bit to your question and the termination issue. First, let's look at E. coli. E. coli is the bacteria. People talk about how bacterial replication works. DNA polymerase 1, DNA polymerase 3. That's actually all from E. coli. There are some differences in different bacteria because, of course, bacteria are a really broad group of organisms. But for basically this class and the whole textbook, when they talk about bacteria, they basically just mean E. coli. So in E. coli, there is one and only one origin of replication in its genome. And the genome is about 5 million base pairs in length, and there are different strains of E. coli, which are all going to be slightly different relative to each other. This is the kind that make you sick, and the kind that we work with in the lab, they're not the same. So if you're ever working in a lab that's working with E. coli, it's not the same one that makes you really sick, and you have to wash your spinach and all that stuff for it. We'll talk more about that later if you're interested. So one origin of replication. From that one origin of replication, you end up with two replication forks. They zoom their way around the genome, and then they bang into each other at the termination site, which is almost exactly opposite. It's called here the termination site. And once you get there, you have a problem if you're the E. coli. And that is when you've replicated to the ends, you end up with these two double-stranded DNAs that are linked into each other. So how do you deal with that if you're an E. coli and you want to make two separate cells, each of them with a genome? Topoisomerase 2, exactly. So you absolutely require topoisomerase 2 for that last step. But at the same time, if you think about these two replication forks that are going around the genome, pulling apart these strands relative to each other. Remember, it says about 5 million. In this case, it's 4.5 by the numbering here. That's the number of turns that you've gone around to get there. And so you also need topoisomerases to deal with pulling apart those strands, because that's the same. Whenever you have replication, you have to have topoisomerases as well. So you also have to have very active topoisomerases. They can be 1 or 2 for that replication. And replication in E. coli only takes about 40 minutes. In some cases, it's actually even faster than that. So it's a really rapid process. So again, you need topoisomerases, two replication forks come together. It's actually quite well known how these terminators work in E. coli. A lot less well known in eukaryotes, in fact. And so that's one of the areas of research of Dr. Crissell here in the biology department, is looking at how termination actually works. So let's zoom in on that initiation set. First, you have specific DNA binding proteins. This is your DNA A protein, binds to, again, the specific sequences. In this case, it's E. coli, right next to ORI, and what I didn't mention before, ORI-C. What does ORI-C stand for? It's the chromosomal origin of replication. You have plasmids, you've got viruses, et cetera, so those have different letters. So binds next to ORI-C. That binding then, through protein-protein interactions, brings in the helicase with the helicase loader protein. Helicase loader protein is functionally extremely similar to the clamp loader. It takes something which is a circle and puts it around a certain number of strands. In the case of the helicase, it's just around one strand. In the case of the clamp loader, of course, it's around 2 strands. And then once you have the helicase loading protein, does its job just like the clamp loader. It's an ATP-dependent process. Putting on the helicase does its thing, and off you go. In eukaryotes, there's something called the ORC. I was going to bring a nice ORC picture from The Lord of the Rings, but I didn't get around to adding it to my lecture. It stands for Origin Recognition Complex. Again, the molecular biologists and their creative naming process. Origin recognition process basically does the same thing as DNA A here. If you look at some of the structures of these proteins associated with DNA, there are a couple of interesting things that you see. The main thing is that they make pretty different structures, actually, in terms of what's happening at the origin of replication. This is what happens in archaea and eukarya. You have a relatively compact set of proteins. There's actually just ORC 1 through 3. There are 6 different ORCs that you have in eukaryotes. In bacteria, the DNA A actually kind of coats the DNA. And then there's some viral examples, because viruses are so cool and different, we have to include them. So once you have this binding to your origin of replication, recruitment of the helicase and the helicase loader, and you have replication. Well, once you've done that, you don't want to have multiple initiation events happening one right after each other. Because if you did, then you're going to have all these replication forks going around your genome, and you're trying to separate the cells, particularly for something like E. coli. How do you make sure that it's gone around once and only once? So that's what's called licensing. For E. coli, it turns out this is very different than what you have in eukaryotic cells. So we'll talk about them really separately here. We've kind of already talked about this process already, and that has to do with methylation of the DNA. And we'll talk much more about methylation of DNA when we talk about DNA regulation, et cetera, later on, particularly in eukaryotes. But in terms of bacteria, on a regular bacterium, a regular happy bacterium, not actively undergoing replication, the DNA is methylated, and it's methylated on both strands. As soon as you have a origin of replication, which fires, it starts, you get 2 replication forks. Now you're going to have hemimethylated DNA, and that's just saying that one strand is methylated, the other strand is not. Hemimethylated DNA in E. coli now no longer will interact with DNA A, so you don't get another round of replication that starts there until after a certain period of time. When else did we talk about methylation in E. coli DNA? Mutations to some extent, but we'll talk more about that later today or on Monday. Code readers, not so much for, that's more for your histones, so for DNA methylation.
New DNA has methyl and old DNA, or new DNA has no methyl and old DNA has methyl.
Right. New DNA doesn't have methylation, and that's actually shown right here, and old DNA does have methylation. Why is that important? Okay, so it was not done yet, but I heard someone say, okay, yes?
So that the proofreading enzyme knows the difference between the old strand and new strand when fixing incorrect.
So basically, I'm going to paraphrase your question. Hope that's okay. That it's the new versus old for error correction purposes. I wouldn't call it proofreading, because proofreading is usually very specific to the polymerase. The error correction that happens because of hemimethylated DNA is the mismatch repair system, which actually is the mute H and L and M protein. So we'll talk more about later today. So it's after you actually have the replication fork gone through. But it's the concept is right on. It's just a slightly different process which is happening there. So it's knowing which is the new strand that you put in. So if there's a mistake that ends up in there, you take it out of the new strand because it's the new one that you put in that has the mistake in it. So that's what the reasoning for the reasoning. It's the process which is there. So both for knowing that you've just started replication there, and also which strand needs to be repaired if the wrong base has gotten put into it. Okay, so let's switch and talk about eukaryotic replication origins, because anybody read the Scientific American where they think that there should be about the same number of bacteria in us as there are cells? So they've gone way down in terms of the number of bacteria rather than the number of human cells. Basically says we don't really have a clue. But I would say 10 to 100 times more bacteria. Well, maybe it's about the same number of bacteria. Varies a bit. So, but yes, we're clearly eukaryo-focused, because we are. So let's talk a little bit about eukaryotes. Eukaryotes have a number of problems as far as replication is concerned. Generally, they've got much bigger genomes than the bacteria, and their polymerases are slower than the bacterial polymerases. That's like exactly the opposite of what you'd expect. Bigger genomes, you're going to have a faster polymerase, right? Well, actually, no. They are slower polymerases. And that's probably because they have to deal with nucleosomes and all of the chromatin structure that's associated with that. So eukaryotic DNA polymerases are slow. They've got to replicate usually bigger genomes. How do you deal with that? You have to have multiple origins of replication. So big difference between E. coli and eukaryotes, multiple origins of replication. And one of the big problems then that you have multiple origins of replication, then you clearly also have to have multiple terminations of replication. So when the replication forks run into each other. And that's another reason why it's a very interesting question about exactly what happens when you have these two replication forks run into each other. Again, we don't have a very good clue about that. How do we know that that's the case? This is a cartoon of the experiment that was done for this. Basically, all that you do is you take a DNA in a cell, You get it to start replicating, and it's convenient if you're working with eukaryotes because they all go through a cell cycle. So you just stimulate the S phase where you get synthesis of the DNA. And then you put in radio-labeled thymidine. Why thymidine? Because it's one of the nucleotides that goes into DNA. It also doesn't go into RNA, because in RNA you have uracil. So it's very specific to DNA. You incubate with tritiated thymidine, which is radioactive, but just for a very short period of time. And then you add a whole bunch of non-labeled thymidine to basically dilute any of that radio-labeled thymidine. So what should happen is you'll have a very short period of time where we'll be incorporating radioactivity, and then a longer period of time will be less radioactivity. It's what's called a pulse chase experiment. It's a very common experiment used in molecular biology for decades now. Once you've done this, now you take the DNA out of the cells, you spread it on photographic film, and you come back a couple of months later. And literally months. You have to wait a very long time because the amount of radioactivity there is really, really small. And then what you'll see, if you look carefully, is you'll have on your autoradiogram, which is that piece of photographic film that you put there that's been exposed due to the radioactivity from the thymidine, you have these little blobs. And these blobs are representative of the replication forks. And what you can see, there are multiple of them spread out across your DNA that you've isolated, and that there's a lot more radioactivity at one spot and less further on. And what that tells you is that the replication fork is moving in this direction, because you had a lot of radioactivity, and then as soon as you did your chase, you have a lot less radioactivity. And so that's what you can see on this particular process. When people first looked at this, they looked in Saccharomyces cerevisiae, baker's/brewer's yeast, and turns out that they're really quite well behaved, wonderful organisms for using for molecular biology. And they were able to isolate what they are called autonomous replication sequences. And we'll talk about these autonomous replication sequences a little bit later on. Basically very much like origins of replication that you see in E. coli or EC, these autonomous replication sequences. You can take these, you can put them on a different piece of DNA, you'll get origins of replication, etc. In yeast, it turns out that between about 30,000 or so, 30 to 300,000 base pairs, you have each of these origins of replication spread reasonably well throughout the genome. The problem is, as soon as you start to look at larger organisms with bigger chromosomes, it's kind of all over the place. You have lots of replication happening in lots of different places and no nicely defined origins of replication. So that's one of the problems, okay? Really long genomes, slow polymerases, multiple origins of replication. But in eukaryotic cells, they all have this cell cycle thing, where through most of the cell cycle, they're actually not actively undergoing replication. It's only during S phase that they're undergoing replication. And even in S phase, for instance, in most human cells, S phase lasts about 8 hours. You can do the back of the envelope calculations. How many origins do you have? How fast are your DNA polymerases, etc.? It turns out that not all of these origins of replication are being used at the same time. You've got some that start early and some that start late, and not surprisingly, euchromatin, which is where you're not as compacted, is where you start a lot of these replication. And then the heterochromatin, you replicate at the very end. And that you can see in this experiment. What people did here is they also added a specific nucleotide. In this case, it's not radio-labeled, something called BRDU, bromodeoxyuridine. And the reason you use BRDU is that they're really good antibodies that you can use to detect it. So in early S phase, you have replication in one particular chromosome at this end, and then it just sort of spreads throughout the rest of the chromosome. And basically, this is just showing that these origins of replications are regulated temporally through the cell cycle and inside the S phase. How that works, we really have no clue. But again, that's something that is happening, and we can see that that's what's going on there. How do people try and study some of these things as well as this labeling approach? A newer technique that people are using for this is something called microarrays. Anybody heard about microarrays? Know what they are? I've actually got one microarray in my lab upstairs in CLSB. Anyone wants to come take a look at it? Basically, all that it is a robot that puts really, really small amounts of very specific DNAs onto a microscope slide. And the current ones can put about, actually I think the latest are about 40 to 50,000 dots on one microscope slide, standard microscope slide, which is pretty amazing. But basically what happens here is you have the whole genome, and particularly areas around what you think is an origin of replication, and you have those sequences, separate sequences, all the way across your microarray. And this is just a really small amount of that. And then you start S phase. After a certain period of time, now you take the DNA, you break it into little pieces, and then you do hybridization. So remember, you can get DNA strands to come apart, go back together on this microarray. And what you'll see is most of the DNA here is just present in one copy. And that's what these yellowish squares are. But some are going to have 2 copies. And that's all that you're doing. You're measuring the amount of DNA which is going to bind to this very specific sequence that you have put down in your microscope slide. And what you see as time goes by, that you get more and more that have 2 pieces of DNA, and eventually, once you've replicated your whole genome, the whole thing has 2 copies. And this is a really great way to identify exactly where, well, not quite exactly, but at least localize to where your origins of replications are actually happening. As I mentioned before, for yeast, they're very specific sequences. For E. coli, it's very specific sequences. For human cells, it turns out it's not very specific sequences at all. And there are lots of possible origins of replication, but only a few of them get used. And this is one of the ways to figure out how that's actually happening. I'm going to talk about one other quick experiment, and this is how people know that either the ORI-C in E. coli is necessary and sufficient for replication, or these autonomous replication sequences in yeast. And basically the way that this works, you take the whole genome, chop it into little pieces, and then add each of those little pieces individually to a gene that the cell absolutely requires to survive. And so that's the case right here. Here's the DNA that the cell needs to survive. This hist gene, it's a gene required to make histidine, and then a whole bunch of random pieces of DNA that get put in here. So here, you take this, you put it into your yeast cells. Most of those cells don't grow in the absence of histidine because they need this gene to make histidine. You get a couple that do grow. We can talk about the exceptions about that later. But if you have an autonomous replication sequence, this gene is going to make lots of copies of that gene once you put it into the yeast. And since it makes copies of those genes, now it can grow in the absence of histidine because it's brought the gene that it needs to make histidine. So by this process, you can find out what these origins of replication are. And then once you've found these little pieces, you can take them, you can put it into another piece of DNA, etc., and get replication to take place. This is exactly what was done to sequence the human genome. People took the origins of replication for bacterial genomes, put them into big pieces of DNA from human cells, got them big pieces, sequenced those big pieces, got another piece, sequenced that piece, etc. And so it was just by knowing about the origins of replication which allowed the sequencing of the human genome. If you look at where those origins are in the yeast genome, you'll see that they're about 30,000 base pairs apart. Not surprisingly, they bind to the origin recognition complex. But here also you have the same problem that we have in E. coli, only more so, because you can't start replication at these origins of replications multiple times in your cell cycle, because if that happens, you're going to end up with this whole cross-linked mess of DNA that's partially replicated, partially unreplicated, etc. So what happens at each of these origins is once you have the firing of that replication origin, i.e. those two replication forks take off and go in opposite directions, then the origin recognition complex stays bound there. But again, just like you have with the E. coli origin of replication, you need to put on the helicase. And the helicase, in the case of yeast and other eukaryotes, is something called MCM. MCM for mini chromosome maintenance. So if you've got one of these things got an autonomous replication sequence on it, you have to have the MCM proteins. If you have mutations of those MCM proteins, you don't get replication taking place. And later people found out that that was what was going on there. Human replication origins are spread apart way more than that. And again, we still don't have a very good clue what makes a human replication origin and how it's actually being used. Again, a very active area of research. But let's concentrate again on what's going on at the origin of replication, and particularly for licensing. Again, just like the case in E. coli, you want to start and only start there once as opposed to starting multiple times. So we've got our origin of replication. The ORC interacts with that. And then we have a couple of proteins here, which are also called the licensing proteins. Again, making sure that you have one and only one round of DNA replication that happens at that particular place. These are the CDC6 and CDT1 proteins. Anybody know what CDC stands for? Genetics? CDC, yes. Exactly. What else does it stand for? Center for Disease Control and Prevention, actually, the whole thing. Cell division cycle. So when you see these gene names and they mean nothing to you, very often there'll be something like CDC. Well, CDC, cell division cycle. Geneticists, as I probably have mentioned before and probably will say many more times in this class, they like to break things and then try and figure out what they broke. And then what they've broken, they'll name the gene after that. So there were a number of now Nobel Prize holding scientists who did this for yeast, and they found lots of mutants that had problems with the cell cycle. And so they just called these CDC mutants, cell division cycle mutants. So one of those is CDC6, and in cell biology they'll talk about a whole bunch more of them. Don't worry, talk about the cell cycle. So CDC6 interacts with the ORC at the origin of replication, and CDT1, which actually is found in a very similar screen as well. Once you have all these guys associated, now you bring in the helicase. Once you've got helicase binding, helicase then gets on one strand of the DNA, opens it up, primase, you've got your DNA polymerase delta, DNA polymerase epsilon, two replication forks take off and do their thing. Okay, well that's great. You get replication going, but now you don't want to start another replication here. How does that happen? Phosphorylation. Our friends the kinases come in, and these are also called cyclin-dependent kinases because the cyclins are proteins that come and go in the cell cycle. Cyclin-dependent kinases that are important for the beginning of S phase will now phosphorylate both CDC6 and the ORC complex. Phosphorylation of CDC6 is the eat me signal, so this gets degraded. And phosphorylation of the ORC complex means that just like with other phosphorylation, what does phosphorylation do? It changes the structure on the outside of a protein. So it's going to interact with different things than it otherwise was interacting with before. So what does it normally interact with in the absence of phosphorylation? CDC6 and CDT1. When it's phosphorylated, it can't interact with CDC6 and CDT1. And above and beyond that, the CDC6 has gotten chopped up and is no longer there anymore. So that's the process where now basically the phosphorylated ORC says, hey, already replicated here. Don't want to do any more. And that's the way that these guys can get one and only one round of replication from each origin which actually gets used. And so the question is, which origins get used? And it seems to be the real critical step is right here, the association of CDC6 and CDT1 with the origin recognition complex. And that's what determines which of these origins gets used when. And that process, again, the part on the up way beyond the top of this slide, is what will hopefully be in the next version of the textbook, because we don't know what's happening at that point. So how do you start? Both bacteria, eukaryotes. The other problem that eukaryotes have is this, these nasty stuff that's all the DNA is bound to, the histones, and then all the other non-histone proteins that are part of chromatin. What happens to these nucleosomes as you have a replication fork that moves through them? Clearly, you're pulling the two strands apart. If you're pulling the two strands apart, the histones are going to be dissociating. You pull the two strands apart, you're replicating. You can't have that with your DNA wrapped around the nucleosomes. So what happens here is you have, the names of these proteins are not important, but the histones come off here and then come back onto the DNA actually right after the replosome has moved through. H3 and H4 stay together. These H3 and H4 seem to be a much more stable complex than H2A and H2B, and reassociate by themselves here. So you've got H3, H4 in blue and in green that have been here, your old nucleosomes. Now you have your new nucleosomes behind. So these guys stick together. Why is that so important? We remember all of our histone modifications that we had back at the last midterm that we've forgotten about already. Those now still have the same modification. So if you've got a modification over here, that modification is going to be present here and here. And because you have histone read-writer complexes, these now will transfer those modifications to the new histones which are being added on the other side. You have to double the amount of histones that you have associated with your DNA. So you've gone from one copy of your DNA to two copies of the DNA. You still got to compact all of that. And what that means is you need huge amounts of histone proteins. And they're in fact some of the most highly expressed proteins in any eukaryotic cell, because you need so many of them. And there's so many of them, and it turns out you need multiple copies of the genes for all the histone proteins as well. So just transcriptional regulation isn't enough to have enough histones around to be able to recoat the DNA after you replicate through them. Also, it turns out that the messenger RNA for these histones is only stable in the S phase, and we'll talk about messenger RNA stability a lot more a little bit later on in the course. So any questions about these origins of replication, extension, nucleosomes, etc.? Yes?
So just to make sure I'm clear, the H3, H4 is the only nucleosome histone complex that is actually remaining after replication. So like when you have the octamer, then I think that's what it's called. Then it's just that H3, H4 tetramer that stays, and then everything else is a brand new quote unquote histone that then has to be covalently modified. Is that correct?
Okay, so the questioning, I'll try and paraphrase here, is basically what's happening with these nucleosomes, and are they completely dissociating new, old, etc.? And maybe I didn't do a good job of explaining this, but basically the H3H4 tetramer, as you say, stays together. It comes off of the DNA and goes back on, because you can't have it there while you have the replication fork that goes through. And so H3H4 stays together, comes on and off. H2A and H2B, those stay as dimers, but they can be mixed and matched. And so that's what they're trying to show in this cartoon. So you have, for instance, this H2H2B, this dark-colored one here, can then associate with a light-colored H2H2B, one of the other ones. On the other hand, you always have H3 and H4. They're always dark or always light. So H2 and H2B, those dimers can exchange. H3, H4 always seem to stay together. Yeah?
So back to the pulse trace experiment.
Yeah.
The BRDU label, how does it fit? No.
This one.
Nice picture.
Oh, the pretty picture that we had here already?
So where does it play into that, if you could?
Oh, so where do you have BRDU incorporation?
Yeah.
Yeah, so if you think about it, unfortunately, this only has like one line for the double-stranded DNA. So BRDU, when you put in BRDU in S phase, you put in BRDU, what's going to happen is it will get incorporated by both the leading strand polymerase and the lagging strand polymerase on the new strand. So the BRDU is going to be incorporated, and so it's being incorporated here, along here, and then in the backstitching orientation, sort of going in the opposite direction along these strands. But it's just basically like new DNA. The new DNA is being labeled by BRDU. And it's the same thing with the radio-labeled tritium. It's going to be that new strand that gets labeled there.
And what does it stand for again?
Oh, bromodioxyuridine. Bromodioxyuridine. That's not critical. We'll talk about deoxyuridine when we talk about DNA repair at this rate on Monday. But the bromo is just so it's a good way to detect it. Yeah.
What's the difference between CAF1 and PCNA?
What's the difference between CAF1 and PCNA? So I'd mentioned you don't need to know CAF1 and PCNA. What is, however, important is that PCNA makes sure that you get all these nucleosomes back together. So as I may have mentioned before, PCNA interacts with a whole bunch of different things. Some people talk about PCNA as being sort of the replication's tool belt. It's got this protein associated with it, that protein associated with it, the other protein associated with it. One of those things in the PCNA tool belt is the CAF1, which is a histone chaperone. So it helps assemble and disassemble nucleosomes. And so we talked about histone chaperones a little bit when we talked about the chromatin remodeling complexes. So those histone chaperones are held at the replication fork with interaction with PCNA. That they're called CAF1 and NAF1 is not important, but that there are histone chaperones that are associated with PCNA, that is. And so PCNA, not surprisingly, lots of people are interested in PCNA and exactly what it does and all these different interactions. Yeah?
So when you talk about the histones, there are a huge amount of histone needed. So
Mm-hmm.
basically it means when the DNA is being replicated, the histone genes is also crazily being
Transcribed and expressed. Yeah, so the question is basically, when are these histone proteins being made, and are they being made at the same time as the DNA is being replicated? And the answer is yes. They are being made at the same time the DNA is being replicated. They're being made in S phase. They're also made throughout some of the other phases in the cell cycle, but because the messenger RNA is not stable, the RNA is made, but the proteins aren't actually being made. Okay, let's finish up by talking about the end problem. And the end problem is also a display problem. And if you printed out the notes, we'll notice that there's actually should be a figure right here. Go figure, pun not intended. So the problem with eukaryotic chromosomes, well, lots of problems with eukaryotic chromosomes, but one of the problems is they're linear. And if you have a linear chromosome with double-stranded DNA, when you get to the end, you still need to make a primer to make that opposite strand. You have to have your primer. And those primer, what makes primers? Primase. Primase makes RNA. That RNA gets degraded by RNase H, and also it's low fidelity anyway, so you don't want to hang on to it. But what that means is every round of replication, the ends of a linear piece of DNA are going to get shorter and shorter and shorter because the RNA primer gets chopped up every time. So what happens is there's a way of dealing with that. That's what telomeres are and telomerase. But a lot of people think that telomeres may actually have something to do with aging. Because if you look at older and older cells, and even cells from older and older individuals, the more times they replicate, those telomeres end up getting shorter and shorter and shorter.
and it is a way of looking at aging, and in some organisms, fruit flies, worms, controversially in mice, if you extend telomeres, you can actually make the cells, certainly in culture, last longer, and in some cases the organisms actually last longer. So there are a number of companies that are trying to do this for anti-aging pills, etc. So there are other things, of course, that happen with aging too. But some people think that this is the answer. Yes?
Was that a selection-based thing where they selected for mice or worms with longer telomeres, or did they actually insert longer telomeres down to the chromosome?
They overexpressed this enzyme, telomerase, is what lots of different ways of trying to do that. But the main one is to overexpress telomerase. And so how do you deal with, if you have replication, and particularly if you're thinking about development, embryos, for instance, they're going to be going around huge numbers of rounds of replication. You don't want the ends to be shrinking every single time. And then you'd have to make them bigger again when you make your germline cells. So you have to have some way of dealing with that. So in actively replicating cells, you have the answer, telomerase. So what does telomerase do? What it does is it adds a repeated DNA sequence to the ends of linear chromosomes. And the way it does that is a really fascinating process. It's a reverse transcriptase, not part of the central dogma. What it does, reverse transcriptases take RNA as their template and make a DNA copy of it. Best known for our friends. Anyone make it to the biology seminar yesterday? HIV? So reverse transcriptase, that's what HIV does. Has an RNA genome that it then uses reverse transcriptase to make into DNA. Here, reverse transcriptase is the telomere. However, the telomerase, has the template for making that repeated DNA sequence with the telomerase enzyme. So it's an RNA, which is part of an RNA protein complex that then takes a 3' end, adds nucleotides to it. And it will add the nucleotides which match the RNA which is present in this RNA part of the telomerase. So it makes multiple copies of that RNA. So what it does is it just extends the 3' end. Just like all DNA polymerases, reverse transcriptases are DNA polymerases too. They'll just extend from that 3' OH. And what happens then is you end up with multiple repeats of the sequence which is in the RNA right here. So that's diagrammed, was diagrammed on here. Why do these slides keep disappearing? Well, it doesn't really matter. But what happens is you end up with multiple copies of this sequence hanging off at the 3' end. Now, all that does, making many, many copies like this, is to provide a template that now the DNA primase can come down and bind to, make an RNA primer, extend it until it gets to the 5' end, which is over here. Because if this particular strand is going from 5' to 3', you can extend this strand, but you've still got to copy the opposite strand back in the opposite direction. Because it was the primer that was bound to this that got chopped off. And to be able to deal with the fact that you keep losing your primers, you've got to make more template to put new primers on. You're always going to have a little piece at the end of your linear genome where the RNA primase was put down and has been removed. So all telomeres actually have an overlap at the end, this extra little piece. But that piece now, because it's been made from this repeated sequence that happens again and again and again here, this single-stranded end can now base pair back over to here because it was a repeated sequence. And so what happens is it loops around and makes what's called a T-loop, which is what you can see right here. So it's a single-stranded piece of DNA at the very end of your genome that then loops around and binds right here. There are also multiple proteins that are associated with this right here, because as soon as you have an end, there are lots of exonucleases in the cell. Exonucleases love to chew in at ends, and we'll talk much more about that when we talk about
DNA repair on, on Monday. There's some really cool reviews giving you way more information than you need to know about this class, but I've given you the links to them right here. There's a whole special issue on telomerases, the November issue last year in Nature. And then just as a review, okay, we talked about the DNA replication semi-conservative, processivity, fidelity, and proofreading, primers and the replosome. It was all in the midterm. We'd been there, done that. Stuff that we talked about today, which is new, or the really initiation of replication. The initiator proteins themselves, and then what happens in terms of licensing, when I say the regulation here, and then the telomeres. Again, I got to figure out why these images are disappearing on me. In terms of that repeated structure, which you end up adding again because it's a DNA polymerase that adds onto the 3' end, will make the template to make multiple copies, and basically sort of fill in the end where your primer was degraded from that lagging strand which you made from the polymerase. We have 4 minutes left. Do you want to keep going or call it a weekend? Call it a weekend.